let us discuss about the transport from the trans-Golgi network towards the lysosome. So normally, as we know, lysosome contains different hydrolytic enzymes. So those enzymes has to be prepared in the endoplasmic reticular lumen. After the production, they are modified using the post-translational modification schemes. After the modification of those enzymes, they are sorted in the Golgi apparatus. After sorting in the Golgi apparatus, they need to be transferred from the Golgi apparatus towards the through the trans-Golgi network to the early endosome. So how they can be transferred? So suppose in this case, in this example, as we can see, is a lysosomal hydrolase precursor enzyme is produced, and it is attached with the mannose residue, and this is the post-translational modification portion from the endoplasmic reticulum. It has been taken, so along with the mannose residue. Okay, so what happened? Addition of N-acetyl glucosamine is done because those mannose residues are very much hydrolytic, so they can be cleaved easily by acid hydrolysis. That's why the N-acetyl glucosamine is actually preventing those mannose residues to be cleaved. So after the modification, it is being carried out. After the carry out of this hydrolase precursor along with the mannose and N-acetyl glucosamine residue is making a signal molecule for mannose 6-phosphate receptor, which is present in the trans-Golgi network here. So this green colored things, this green huge receptor are called the M6P receptor or mannose 6-phosphate receptor. These mannose phosphate receptors are there, to which this hydrolase precursor molecule is attached via the mannose linkage residue. After the attachment, clathrin molecule comes in, they coat the whole thing and they pinch out one thing and finally make a vesicle of it. So this vesicle, receptor dependent transport procedure via the receptor dependent transport machinery is being carried out from the trans-Golgi network towards the lysosome, which is called the early endosome state of the lysosome. So this vesicle comes in and it will fuse with the early endosome, and it will eventually gonna release this precursor molecule of hydrolase. But this release needed to have the acidic environment. So without the acidic environment, this release cannot be established, or this dissociation cannot be established. But as we know, dissociation is really important because we need to recycle this receptor molecule from the early endosome towards this trans-Golgi network, because cells will need to go through this cycle again and again. But for doing this, we need lots of hydrogen ion concentration inside the early endosome, and that can be established via placing a proton pump in the membrane of early endosome. So we can see a proton pump present there, deriving the energy of ATP hydrolysis. The proton pump is going on, and the hydrogen ion from the cytosol inside the early endosome can be pumped in. And after the pump establishment is done, the hydrogen ion concentration inside the early endosome goes up, and that means the acidic environment is gonna dissociate this protein molecule, this hydrolase precursor molecules, and also it will cleave these mannose residues. It will cleave this N-acetyl glucosamine residue. It will cleave those residues. Okay, and it will remove some phosphate groups from the mannose 6-phosphate residues, and after the removal of the phosphate groups, it will finally fold properly and produce the hydrolase enzyme. So this is now the activated hydrolase enzyme inside the early endosome, which is gonna in turn make a proper enzyme inside the lysosome, because this early endosome is gonna mature into and produce into the lysosome. Now after dissociation from this receptor molecule, the receptor along with other retromer coat proteins are there. They are becoming free, and the coat proteins or retromer coat protein comes in, and not clathrin. You can see there are no clathrin to go towards the trans-Golgi network from endosome. So in this case, retromer coat is done, and after the coating, a part of this membrane will be bulged out along with the receptor for the mannose 6-phosphate, and finally it will make a vesicle along with this mannose 6-phosphate residue, and finally it will come and the mannose 6-phosphate receptor comes in, and it will attach with this trans-Golgi network again, and it is ready for going through another round of this process. So that's how the process will go on. And the all process is one starting point, one destination point, and inside and in between the transport of vesicle. So vesicle, and I call it vehicle, all these things are same. So vesicle means vehicle. Again, production of something, destination point, the journey from production point towards the destination point via the vehicle system. That's how all this thing is done. But one very very important thing in this case I must say is about this proton pump, because this pump has to be active all the time to keep this acidic environment inside the lysosome really stable. So it is not only needed for the dissociation of these precursor molecules from mannose-6 phosphate residue, but also it is important for the acid hydrolysis, which is a very very important work of lysosomes. That's hope, and I hope that can help you.